Good morning, good afternoon, and good evening to everyone joining us from wherever you are around the world. Distinguished guests, ladies and gentlemen, it is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with our main presentation, allow me to provide a brief overview of the initiative that brings us together today. Instal Africa, Innovation, Science and Technology for Action and Learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. Instal Africa was born from a collaboration between the Foresight Institute of Research and Translation Rwanda, or FIRAT Rwanda, and its technical partner, Drosophila UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. With that foundation laid, it is now my distinct pleasure to introduce our invited guest speaker.
Ladies and gentlemen, it is our great pleasure to introduce our distinguished guest speaker for this session, Dr. Hamed Badmos. Dr. Badmos is a cancer researcher and a leading advocate for the use of Drosophila melanogaster as a powerful model in both fundamental and translational research. He currently serves as a postdoctoral researcher and Chief Scientist Office Fellow at the University of Glasgow's Wolfson Wool Cancer Research Centre, where he harnesses the advanced genetics of the fruit fly to better understand cell signaling and cancer progression. His academic journey began with a degree in biochemistry from the University of Ilorin in Nigeria, and continued with an MSc and PhD in Advanced Biological Sciences from the University of Liverpool. His path reflects the global impact and excellence of African scientific talent. Dr. Badmos's research has generated important insights into collective cell migration, a process essential to embryonic development and cancer metastasis. During his doctoral work, he identified the D-ubiquitinase non-stop, also known as Drosophila USP22, as a key regulator of the actin cytoskeleton during border cell migration, offering new understanding of how tumors invade surrounding tissues. He has also contributed to uncovering the role of ataxin 7 in regulating cell signaling pathways such as Hippo and YAP, further demonstrating the relevance of Drosophila in studying conserved biological systems. Currently, his work integrates medicinal chemistry and computational modeling with high-throughput Drosophila screening to develop novel therapeutic strategies for colorectal and pancreatic cancers, bridging the gap between fundamental discovery and clinical application. Beyond his research, Dr. Badmos is deeply committed to advancing science in Africa. He is the Nigerian pioneer of the Droso for School initiative, promoting the use of Drosophila as an accessible tool for science education and research. Through his involvement in Drosso Africa workshops and mentorship of students, he continues to play a key role in building scientific capacity across the continent. A recipient of the prestigious Tenevis Scotland Grant and the CSO Scotland Fellowship, Dr. Badmos exemplifies how fundamental research can drive innovation, inspire communities, and shape the future of science in Africa. Dr. Badmos, we are honored to have you with us today. The floor is yours.
All right. All right. Thank you so much for the invitation and thank you so much for the introduction as well. So that was detailed. So I'm not going to include any more introduction into my slide. So today I'm going to talk to you about the use of Drosophila in cancer research, and I'm going to elucidate, why we use Drosophila and some key features in using Drosophila for this kind of research. So I'm going to be guided by the following outline. So I'm going to show you what models we've got in biomedical research, the models in cancer research, why we use Drosophila for for research and as a model organism, and also the use of Drosophila in cancer really. Right. So introduction into models in biomedical research. So biomedical research is actually a complex research field and requires several models. So before we actually dive into biomedical research, let's go back into the history. So the use of models actually came from the 19th century that, scientists, for example, Claude Bernard, a French physiologist, was looking to to study experimental medicine. And then it was like, what should we use? We can use patient, we can use humans. So then he proposed the use of, various species of animals to actually study experimental medicine. So including, infectious disease, including tuberculosis, including, malaria and the likes. And then then looking for animals that can allow us to study this problem conveniently, because you can, for example, you can use like wild animals to to study things, because they're quite dangerous. So we need something that is amenable to changes, something that we can work with easily, something that preferably small, and something that is going to be quite cheap. So, and this became the August Croft principle in 1975. So, this guy is a Danish physiologist, and also he perfected the use of models in research. Right. The German British scientist, so Hans Krebs and his son John Krebs, and guys, people that are biochemists will know that these are pioneers in the biochemistry field. So they caution against unnecessary extrapolation, because it is easy for scientists to be like, okay, I've studied this thing in like mouse model, and oh, then that is what is going on in patient. So we need to be conscious and cautious of extrapolation from one species to another, based on the fact that we've got different anatomical changes in this species. Although the physiology might be similar, but for example, in flies have got wings, we don't have wings. So if you actually study something in the fly wings, you can extrapolate that easily to patients. And also we should avoid crude generalization. So we shouldn't think that, oh, because flies can fly, we humans can also fly as well. Or because flies have got tumor, patient can also can have tumors, or because patients have got tumors, mouse models can have tumors. So we should always we should be conscious of this generalization and avoid crude generalization. Right. So I'm going to dive into different types of model. So, and this was actually put together by Ransom in 1981. So he actually divided model into two types. So the first type is the descriptive model. So this is a model that allows you to describe a process. And I'm going to give an example of this type of model in my next slide. So allows you to describe a model. It could be an existing model that you already know, but you want to find a simpler way to actually describe the model. So a descriptive model would allow you to do that. We've got a discovery model. So a discovery model allows you to discover new process, allows you to discover how a process works, allow you to discover, better understand a process, and also, come up with new process really. So those are the two types of models we've got in science. Right. So descriptive model and discovery model can then be divided into various type of models. We've got mathematical models. So this is when you've got a mathematical equation that allows you to describe a process. So for example, force, you want to describe, what is force. You can write a mathematical equation for that. You want to describe the speed of light, you can write a mathematical equation for that. You want to describe like walk. So what would be the formula for walk? So these often model analogies. So the quite analytical and the the modeling a process and the we refer to as modeling analogy. So we've got computational models. So computational models can be in form of animation. So let's say you want to describe, cell migration. You can describe this using a computation model that, we've got cell that will migrate from one point to another. The cells can interact with the environment. It can interact with some ligands. It can it can require energy. It can require nutrient. So you can model that using computational model. And also that's a model of analogy. So another model, and this model also describe a process, but we can also discover a process, and thanks to the likes of artificial intelligence now, so you can use that, you can feed in information into that with the hope of then using that as a thinker to then come up with a new process and use it to really discover a new process. So we are lagging behind in using AI to actually discover new process, because what AI is doing right now is feeding off of what we've got and not necessarily discovering something new, but perfecting what we've got right now. So I think it's still in the early days to use AI to discover, but I'm optimistic that the future is bright for that. So also this is a modeling of analogy. And we've got substitute models. So these are mainly living organisms, and they can be used to describe a model, a process, and it can be used to discover a new process. So we also refer to them as a surrogate, right? So let's say you want to discover, the cause of cancer. You need a substitute model to do that. So because we can't do that, efficiently in patient right now, so we need a model to do that. Let's say you want to discover, the cause of diabetes, right? So you need a model to do that. So these are substitute model, and you need that model to be living. So this model, then they can model for both analogy and also homology. So analogy is really, you're modeling the analytical process of a thing. You're modeling, how that thing happened, and you don't need to have similarity between your model and what you're trying to model. So basically, that's a model of analogy. But model of homology is you need some similarity. So let's say, for example, you want to study, a disease of the guts, a disease of the guts that affects, patients. You need something that is similar to that, so that something that has got guts, so that you can then study that, that process in the gut of your substitute. Right. So that is, those are the types of models that we've got, and group them into descriptive and discovery process. Right. In biomedical research, we've got several types of model. So these models allows us to answer critical research questions. So let's say, for example, you want to study, let's say, photosynthesis, right? So how plant generate their their nutrient. So you can begin to think of, so what model should I use to study this? What environment do I need to study this, right? So, or you want to study digestion, our food pass from our from our mouth to to our colon. So you can begin to think of, so what model should I use to study this, right? So you need to have a research question, and then think about the best model to study your research question. So if you're using a random model to study what to study your research question, then you're gonna get garbage out of it, really. So you need to make sure that you're finding the right model to to study your research question. Right, and in biomedical research, we've got different types of models. So we've got computational models, we've got mathematical models as well. So for example, you might have heard of AlphaFold. So AlphaFold, based off of existing data of protein structure, of protein modeling, that can then allow you to predict new protein structure, even things that don't have structure right now. So even if you don't have the money for like X-ray crystallography or cryo-EM, you can predict the structure of your own protein that you're working with. So that is really a computational model. You can write a mathematical model for, let's say, the impact of an inhibitor on on your gene of interest or protein of interest. So you can begin to be like, oh yeah, so when we've got inhibitor in the system, what happens to this protein activity? And when we don't have inhibitor in the system, what happens to this protein activity? So and you can feed that off your own existing data. So we've got, so we've got computational and mathematical models. We've got in vitro models. So these are models in the glass. So example of them includes human cell lines. We've got patient-derived organoids, which I'm going to allude to in next slides. We've got bacteria. So mainly these are not necessarily, they're not living, but what you're doing is your experiment is confined into a space. It's confined into your test tube, it's confined into your flask, it's confined into your petri dish. So really, those are in vitro models. And we've got in vivo models. So these are mainly living things. So things that not necessarily, confined to a glass, but they've got their own free will to some extent, not to a large extent, but to some extent. So these are like mouse models. We've got zebrafish, we've got Drosophila. So these are in vivo models, and these two models, both the in vivo and in vitro, allows us to understand fundamental biomedical research. So, for example, bacteria are being used really well for like protein crystallography. So we can identify new protein structure using bacteria. We can identify new therapeutics using human cell line. We can identify, how let's say, for example, developmental biology using zebrafish. We can understand genetics using Drosophila. So these are really powerful models that we continue to use in biomedical research. And in cancer research, we've got several types of models. The first one I'm going to allude to are mouse models. So for you can create mouse model in two ways. One is that you can clone your gene of interest into a cell line, or for example, a cancer cell line, and then you can inject that into your mouse model, and then you can treat that with a drug of interest, and then you can measure whether is the tumor growing or not, and then you can measure the impact of your drug of interest as well, and then you can dissect out the tumor and look out for metastases, or have you impeded metastases, have metastases improved or increased as well. So these are things that you can use this type of mouse model for. So another type of mouse model is a mouse model that is genetically engineered. So like what we do in Drosophila, which I'm going to allude to later on, is that you can clone things into mouse, and then you can allow them to express a particular, let's say, gene of interest. So let's say, for example, that, oh, these two genes are causing cancer in patient. You can then induce that in your mouse model, and then you can ask if they actually, if that combination actually induced tumor in the mouse model. So those are called genetically modified, mouse models. And when you inject cells into them, those are called, xenografts basically. So these mouse models, these are a key tool in cancer research, and people continue to use them, and they've been really versatile in understanding how cancer work and also the discovery of new cancer drugs. Like so, zebrafish has been a fantastic model in developmental biology, and now people have started, and this is really the clinical trial is going on right now, that people have started shuttling cancer cells derived from patient into zebrafish, and then using this to screen for compounds. So what they are doing is similar to what we've done in flies as well, that they're shuttling these cells into zebrafish, and then they're repurposing compounds. So because if this is in clinical trial, you can use this for new compounds. You have to repurpose, set of existing compounds, so that you can then prescribe these to patients. So that shows you that zebrafish is also contributing right now to cancer therapeutics, and I'm pretty sure the clinical trial have started already. Note that this started in Portugal, so I'm not sure if it's been moved to other countries yet, but as of now, this is going on in Portugal. Right. So from the 2010s, so mouse models have been around, I would say, since the 50s. Zebrafish has been around the seasons, since about that time as well, as especially as a developmental biology model. But patient-derived organoids have started to make their impact from the 2010s, and the meaning of that is that you can derive a patient version of a cancer model in the lab. So how would you do that? So what we routinely do is that we go into patient, thanks to the clinicians and the pathologists, and then they can give us a part of the patient tumor, because they're surgically removing them anyways. So, and they're quite useful for research. So what we can do is we can then digest this tissue from the patient, and then we can isolate the cancer cells, and we can grow them up in our lab, and then we can use them for a variety of things. A lot of them won't survive. A lot of them will form what we'll call patient-derived organoids, and then we can continue to passage them. We can what we call, we can preserve them in the freezer, so that we can have more of them. In addition to that, we can use them for various things, including genomics, transcriptomics, proteomics. So, for example, here, what we can do is we can stain for protein of interest. Oh, what happens to this pathway in this sample? And in addition to that, is that correlating to patient? So, because a lot of the time now, when patient goes into the clinic, they can donate the cancer cells, and then that can directly on its own undergo what we call genomics analysis, and then we can identify the genes that are actually causing this patient cancer, and then we can compare that to what we've grown in the lab. That is the PD, the patient-derived organoid, and then we can begin to, like, oh yeah, is this patient-derived organoid, is it a surrogate for this patient here? So, and we can begin to screen for compounds. We can begin to repurpose existing compounds. We can begin to use it to develop new therapeutics as well for this patient. So, and this has been really useful, for example, in cancer so far. And people are also using this in other type of disease, including neurodegenerative disease as well. Right. So, introduction to Drosophila. So, why do we use Drosophila in cancer research? So, as a model organism, actually. Right. So, we use Drosophila as a model organism because they've got, at the system level, they've got systems are quite similar to human. So Drosophila have got, nervous system. We've got nervous system. They've got a digestive system, and we've got a digestive system. There are also some anatomical differences, but the physiology is quite conserved. And in addition to that, 75% of genes that are actually causing disease in human, they're also found in Drosophila. So basically, we can study these genes, in Drosophila, and you know and consciously, extrapolate, or courteously, extrapolate to patient. So, and in addition to that, we can begin to understand, this process. And for example, if it is a disease process, can we drug this process as well in flies, and can we use that, as a surrogate for patient? And one need to be very careful when doing that, that extrapolation. Right. So the contribution of Drosophila to cancer research so far has spanned through 100 years, and the first person that actually recognized tumor in flies, she's called Mary Stark, and she identified that, oh, actually flies can bear tumor, because cancer has been around since about that time as well. So that, actually, fly can bear tumor. And Liz Gattav actually identified the first fly tumor suppressor, and from then there were other, tumor suppressors that have been identified, including also, we know that cancer can be driven by two types of genes. So the loss of tumor suppressors, and the activation of proto-oncogenes, or the overexpression of oncogenes. So there are also oncogenes in flies that, that have been identified, and found that they are conserved in patients as well. And the first, fly model for cancer metastasis were actually identified by Chen Shu and Elena Richardson lab. So, and they were like, combining two type of genes together actually induces metastasis in flies, and that is akin to what we can find in patient. And Ross Kagan was actually the first person to actually cure, or to treat, fly models of cancer. And in addition, although the goal wasn't to cure flies, but that's just the starting point for us to then move into patient, and is a very solid foundation for us to move into patient. And I've only highlighted few labs here, but there are several other labs that have contributed to our understanding of Drosophila and cancer, over the past 100 years. So another power of Drosophila, and the reason why we use Drosophila is because Drosophila is quite cheap, so we can get easy access to it. We've got really we've got strong genetic tool in fly, so we can modify things easily, and also we can model things easily, so we can image them, so we can do several types of assay on them, including imaging. And not only that, but also we can quantify these images as well. So for example, you can see two types of videos going on. So one where cells are rotating and then not necessarily migrating as much, and we can quantify this rotation. And in addition to that, we've got another video on the right where cells are, they're migrating, they're dying, and development is actually going on. And also, we can measure this process. So a question you can begin to ask is, okay, let's say we remove a particular gene that would induce, cell migration in this system. Will the cells still migrate? The one on the left that are rotating, let's say we induce something that will prevent them from rotating. Will they still rotate? And how does that extrapolate to human? Is that really relevant to any human disease? Is that relevant to something that is outside cancer? So those are the questions that as scientists you can begin to think of, and begin to use this type of models to answer. And also, I need to mention that the community in Drosophila research, they're really, really good community. I'm part of a variety of community now based off of my research. But I think Drosophila community are actually the best so far in terms of sharing reagent, in terms of sharing expertise, in terms of actually listening to your ideas and trying to to contribute to your ideas. So being part of the Drosophila world, I would say it's been a rewarding thing for myself. Right. So Drosophila models of cancer. So I'll touch on, I'll say, several models, and this is going to be a bit of a review over the years of what people have done and how they've modeled different aspects of cancers in flies. And I've not mentioned this, but cancer is driven by several hallmarks. And one thing that you want to use your model for is, can you model all the hallmarks, which I think would not be possible, but can you model part of the hallmarks? So one of the hallmark of cancer is they're resistant to cell death, for example. They've got their own blood vessels, for example, angiogenesis. They've got tumor proliferation, for example. And they're driven by tyrosine. They're driven by receptors as well that allows them to to divide and then be resistant to therapeutics, for example. So and also communicates with the environment, for example. So we've got various hallmarks of cancer. So we are using Drosophila to model some aspect of these hallmarks. In addition, we are also using Drosophila to model the pathways in cancer. So there were known pathways in cancer, for example, the KRAS signaling pathway, for example, the P53 pathway, for example, the EPO signaling. So we can also use Drosophila to model some aspects of this pathway, because we know that this pathway contributes to to cancer development, cancer initiation, cancer progression, and also cancer drug resistance. So we can begin to model these processes or these pathways in cancer, in addition to the hallmarks of cancer. Right. So what we've done in the past is that we've modeled tumorigenesis in flies, and we know that, for example, in the control on the left, it is clear that the structure of that tissue or organ is single layered. So it is single layered. So, and that is similar to what we can find in tissues, right? So our tissue has got nicely organized apical to basal polarity. So that's the organization of our tissue. But on the right, we can begin to see that when we actually mutate a particular gene called ataxin 7, we find that rather than that single layering of the epithelium, they actually have multi-layering. And you can see that some cells are actually migrating into the environment as well. So here we've used Drosophila to model an aspect of cancer. So question you can begin to ask is, okay, you know what? Can I treat this process? Can I treat this tumorigenesis? And is that relevant? So you always need to ask, is that relevant to patients? Do we have patients that have got mutation in ataxin 7, that anything that I'm discovering is going to be useful for them? So those are the questions that you need to begin to ask and answer as a scientist that is trying to use Drosophila as a model. Right. So also modeling tumorigenesis here, people have combined what we call, oncogene, so with what we call a tumor suppressor. So here they've actually looked at the larva eye, really, so eye antenna disc. So and then what they've done is, in the wild type, we can label things with the green or GFP. So in the wild type, the eye is actually normal, and then when you overexpress what we call RAS, so RAS is the most mutated oncogene in cancer. So it's mutated in about 40% of overall cancer. And for example, in colorectal cancer, that increased to about 50%. So yeah, so is a known oncogene in the cancer field. So when you then overexpress that oncogene, There's a bit of overgrowth, but really not that dissimilar from the wild type. And then there was this thing called a tumor suppressor. So a loss or the absence of that tumor suppressor actually does not lead to overgrowth, because the tumor suppressor is not there. But still, that doesn't really lead to any significant phenotype, except for reduction in the size of the eye of the eye disc. Now, and then when you combine the loss of a tumor suppressor and then the gain of that oncogene into the animal, you can begin to see the striking phenotype or the striking change in the eye of the animal. And also, this is looking at the overall larva. But when you then dissect the eye out, you can begin to see that that is really massively, overgrowth. So this is similar to the bumps that we see in, for example, in cancer patient, and then that can be surgically removed. But if it is in a dangerous place that you can surgically remove it, then you need to treat it. So this type of model could be an example of what you can use to screen for compounds that will allow you to treat, those type of tumor. Right. So another thing we can use fly for is actually tumor transplantation. So here, when you actually inject like normal cells or normal, with cells that haven't got any disease in them, into flies, you can begin to appreciate that they fail to grow. But scientists have then injected these two genes, so RAPS and MIRA. So these are genes that are required for genome stability. So, and their mutation leads to genome instability. So basically, what you can begin to do is you can begin to cramp up, cell proliferation once you inject this mutant into your animals, and then that leads to invasion into various part of the animal, including the ovaries, including the gut and everywhere, and even and far and distant sites as well. So because they've injected this into the abdomen, so you can begin to use this to also screen for things, either compounds or even genes that will prevent this type of tumor proliferation. Right. So another thing that we can use Drosophila to model is we can use it to model, nutrition basically. So here people have used the wing of the animal, and then they've overexpressed what we'll call EGFR. So is a receptor tyrosine kinase that is part of the cancer pathway that I mentioned earlier on. So, and then that leads on its own leads to overgrowth. But when you also feed these animals with sugar, there isn't anything going on. But when you combine EGFR activation and the activation of a glucose transporter, you can begin to see striking phenotype. And in addition, when you combine the EGFR with sugar, like high sugar diet, you can begin to see striking phenotype. But the biochemistry, part of this, and what is really fantastic is, in the glycolytic pathway, we've got LDH that is literally almost the last phase of that pathway. When you then remove the lactate dehydrogenase in the system, you can begin to rescue the phenotype, right? So basically, this is so it's showing that this tumor is dependent on the glycolysis pathway, and you need to shut out that glycolysis pathway for you to actually treat the tumor, showing you the power of using genetics and biochemistry in identifying, could be novel pathway in cancer, and or better ways to actually treat cancer. So what you can then do is, for cancers that are dependent on, let's say, high sugar, you can begin to like, maybe we need to have an LDH inhibitor that can help to treat this cancer. Right. So now we're modeling various aspects of cancer. We're using various part of the animals to do that. But remember that, for example, these are not necessarily specific. So now people have started looking at, okay, can we model specific cancer in flies? So for example, here people model colorectal cancer. So what they've actually done is APC is the most mutated gene in colorectal cancer. So what they've done is they've looked at the loss of APC in the animal gut, compare it with the wild type, and seen that, there was some tumor, growth compared to the wild type. In addition to that, RAS is also very well mutated in colorectal cancer. In addition, they also saw that, actually after 4 weeks, there is a bit of overgrowth with RAS alone. And then now they can combine the two together and find that, after 4 weeks, they can see really massive growth of the gut. So what they can then use this for is, because, this is really, a colorectal cancer model. They can, for example, repurpose existing drugs that are being used for colorectal cancer and ask if they can use this to actually treat the patient as well. So, and showing you the power of having something that is quite anatomically similar to the disease of interest that you're trying to study. Right. So another model that we've got is a model for medullary thyroid carcinoma. So this is another type of cancer, and the beauty of this is being driven by mainly one gene. So the name of that gene is actually called RET. So when you overexpress RET in the fly eye, you begin to see rough eye phenotype. And the beauty is then you can then screen for compounds, and you can then rescue that phenotype. And it is of interest that this compound actually led to the approval of vandetanib, that is actually used as a first treatment for medullary thyroid carcinoma. So although the people, so actually this came from AstraZeneca. So AstraZeneca already did cell experiment with this drug, and they're like, oh, we think it's working. So but they need an in vivo validation, and then Drosophila was used for that validation. So showing how collaborating even with pharmaceutical company can help us understand or identify new therapeutics in academia. Right. So another thing that we can use Drosophila for is using it as a tool for genetics and drug screening. Right. So here, what we've done is we've actually express things in the Drosophila wing, in adult wing. So let's say we use that red that we've used earlier on, we express this in the wing of the animal. We can see that there is irregularity in the wing, right? So so then what we can do is we can then ask, oh, what happened if we remove a copy of a particular pathway, or kinase? Can we rescue that phenotype? And also, if we add a compound to that, what happened to the phenotype? And what you can then do with Drosophila is you can begin to combine your genetics and your compound, right? So now, if you combine, the remover of that kinase called ERK, and then AD58, that is a compound of interest, you can begin to reverse this phenotype from this to this, which you can see from that is so that is showing you the power of actually using different type of toolkit in Drosophila research to help us better treat our disease of interest. Right. So another power of Drosophila and the use of Drosophila is in personalized medicine, and this has been pioneered by our lab in the Kagan lab. So what we can do is, I showed you earlier on that when people are generating the patient-derived organoid, the patient tissue itself goes into genomic analysis, right? So we can begin to identify the genes that are required for this cancer. So what we can then do is we can begin to group these genes into the driver mutation and the passenger mutation. So the driver mutation are often required for the cancer initiation, they're often required for the cancer progression. So what we can then do is we can shuttle those genes that are important into flies, so to create an avatar for the patient, and then we can use this to screen for compounds, quickly screen for compounds, and then prescribe that to the patient. So that is really the pipeline, and that led to the development of a company that is down in London called Vivan. So basically, what they do is literally this. They can go into patient, sequence the genome, and then try and come up with compounds for them using their own matched fly avatar. Right. So that's the beauty of using Drosophila. Now, what we are doing now is, we're moving from that, and then we are thinking of, the aspect of personalized medicine, because you really need that to be fast. You need to repurpose compounds for that. So we are thinking of then coming up with new compounds. So we are using kinase inhibitors. So vendetinib that I showed earlier on, although it's being used for medullary thyroid carcinoma, so we are thinking of, can we repurpose it for colorectal cancer as well? So, and then we combine. So the kinase inhibitor are divided into different parts of different arms. So the black arm is referred to that part that actually binds to the ATP binding side of kinases. So because these are kinase inhibitors, and also we've got a cap domain that actually binds to, that confers specificity on kinases. So what we can then do is we can begin to tune this compound to confer colorectal cancer activity on it, right? So that was the goal of this project. So what we can then do is we can combine, our structural understanding of the compound with chemistry and a bit of chemical biology as well. And in addition, here we've used a system called computer, that is a robotic chemistry. So this allows us to synthesize similar compounds with simple changes in them in high throughput manner. So then we can bring in our models. So our models include flies and organoids, and then we can use them, especially flies, to quickly screen for compounds that would show better efficacy than vendetinib. And I need to mention that vendetinib, I've got zero activity in colorectal cancer models, and we've tested and we've seen that. So now that gives us a room to actually improve vendetinib for colorectal cancer. And I'm happy to inform you that then we found this compound called AP443, and this is really simpler than vendetinib, although it's got better activity than vendetinib in colorectal cancer, and also keeps its multi-targeting activity, so target multiple part of the colorectal cancer pathway, and these confers efficacy for these compounds far better than what we're seeing with vendetinib. Now, and that brings us to the field of agentic chemistry. So now we're in the world of artificial intelligence. We're in the world of a lot of language models. So what we can then do is, because Drosophila are quite cheap, quite fast. So what we can do is we can combine our Drosophila genetics with agentic chemistry, in the sense that we can begin to be like, okay, we've got robotic chemistry there. We can also modulate our flies, using robotics as well. And then we can begin to explore complex chemical space using our agentic chemistry. So, and then we can have a loop, basically. So without a scientist in the loop, we can predict new compounds. We can quickly test that compounds on flies, and then we can use that to learn, and then we can go back to the synthesis again. So that is really the future right now, and that is what we are pushing forward to do in the next few years, really. So showing you how Drosophila can always be compatible with whatever research question you're trying to ask, whether it is computation, whether it is medicinal, whether it is chemical, whether it is biological. So you can begin to fit in Drosophila into several of your research questions. Right. So far, I've shown you, models in biomedical research. I told you about the models that we use in cancer research. I alluded to why we use Drosophila as a model, and also the use of Drosophila in cancer. And I will appreciate you for your attention. Thank you so much. So I'm going to stop sharing my screen now.
Thank you so much, Dr. Badmos, for that insightful, educative presentation there. We all know
Thank you.
that
Thank you.
cancer remains a challenge, and advancing research, educating us on recent models, the way forward. These researches are driving innovations and is giving us hope for the future, especially in low and medium income settings. We have so many challenges. We have been exposed to so many diseases, and research work, they're creating this avenue that's telling us that in the next years, it's going to get better. So at this point, the audience, we move to the question and answer session. If you have questions, you can put it in the chat box or you can raise your hand. Okay, all right. Thank you so much. We have a question here that says, thank you, Doc. You mentioned bacteria as an in vitro model, but fungi as in vivo. Kindly elaborate. Thank you.
Yeah, no, yeah. Thank you so much for that. So that was a very, very good question. So yeah, so there is a bit of because bacteria they're mainly prokaryotes, right? And then fungi are eukaryotes. So that's number one. So another is that because when we actually grow bacteria in the lab, although they're being grown in like glass, really. So we routinely use them for for modification of things. So, for example, let's say you want to study a particular enzyme and you want to synthesize that enzyme. You use your bacteria to do that. So we can regulate them just in the flask. Is that okay? But for example, fungi they are quite higher in the hierarchy compared to bacteria. So that's why we still refer to as fungi as as in vivo compared to like in vitro. And also fungi has got several several things that that we can use them for as well, in addition to to purifying protein, in addition to even studying development. So we can't really really use bacteria to study development because they're single cell, and that's it. But we can use fungi, for example, to study development. People use them to study cell migration as well. So that is it is just because of the hierarchy. That's why we group them into either in vitro or in vivo. Really.
Thank you so much for that. So we go to the next question and says, recently there has been studies of the tumor microenvironment and microbiome. A very good example is CRC. How can one use Drosophila for study of cancer and the microenvironment?
Yeah, this is a very, very good question. And I would say, people have started, they've started using, Drosophila to study this type of question, and you're bang on with that question. So an example of where you can do that and how people are doing that right now is they can create a fly model that haven't got any bacteria at all. So they can cleanse the system of the animal. Is that okay? And then what they can then do is they can then feed this animal with a particular strain of bacteria, and then they can induce tumor as well. And they can begin to do, for example, sequencing the gut of this animal to ask, how are the bacteria interacting with the tumor. In addition to that, they can vary the type of bacteria they're using by asking, oh, what bacteria will induce, tumor progression more than the other. So people have started to do this. And in addition, even there are people that are proposing that we should feed cancer patients with antibiotics. But I think that wouldn't work because of we know that antimicrobial resistance is something that is real, right? So, and multi-drug resistance is something that is real. So feeding cancer patient with antibiotics would induce things like that. So we don't want to do that, but we want to use the microorganism or the or the bacteria to understand tumor progression. And what we can then do is we can think of new ways to dampen down or to enhance the bacteria. Depends on what you're doing, really, because there are good bacteria, there are bad bacteria, right? So depends on what you're doing to help understand drug discovery or the disease progression.
Thanks, Doctor, for that explanation. And that brings me to my own question. In an African setting, we have challenges on funding, but we have abundance of knowledge and very good researchers. So can we say that this Drosophila can be used for very top tier research works despite limited funding?
Yeah, this is a very, very good question, and I'll say yes, capital yes to that. And and the first person that came to mind, for myself is Professor Amos Abalaji is in the University of Ibadan in Nigeria, and he's actually developed a new center that is looking at Drosophila and using Drosophila as a model. So and he's actually we've got collaboration together, and he's doing really fantastic work. And to me, that is a very good example of the use of Drosophila from a limited resources environment to actually do things that are fundamental. And I would advise that anyone that is actually interested in using Drosophila and trying to build it from the ground zero to to really a very top notch research tool or research environment, he is a very good example of someone to contact, and I'm sure he's going to be more than happy to guide. So yes, we can use Drosophila to do top-notch research. And in addition to that is that because Drosophila is cheap, and okay, so actually what Abalaji and colleague has done is that they've actually improvised as well. So there were things that we buy for like let's say 25k pounds here. You can repurpose or you can get it for like £1,000 over there. Depends on how you modify and improvise and come up with new ideas for that. If that makes so yeah. So you always can do good things, but it just requires a bit of thinking and talking to the right people.
All right, thank you very much for that response. So move on to the next questions. What do the mutations in genes like ataxin and RAS, etc., cause in the pathways they are in? Do they misfold or increase or reduce activities?
Sorry about that. The alarm is gonna go off just for 1 second. Yeah.
Okay.
Okay. So yeah. So ataxin is actually referred to as what we call a ubiquitinase. So and what that does is it removes ubiquitin from protein of interest, so that prevent the protein from degrading. That's ataxin. Not very, very popular. So I'm not gonna bore you with the details of ataxin. But KRAS is RAS is a very, very popular gene. So we've got three different types of RAS. We've got KRAS, NRAS, and HRAS. So and KRAS is actually the driving factor in the RAS signaling. So the pathway is part of things that modulate cell proliferation. Okay, yeah, so we are back. So that should be fine now. So yeah, so KRAS actually modulates, cell proliferation, cell growth, and other cell signaling pathway. So the mutation in KRAS leads to activation of those pathway. If that makes sense. So because it's a proto-oncogene, so mutation in it will lead to oncogenic phenotype, and part of oncogenic phenotype is activating pathway. So, and then now what we do and what we target is we target either upstream of RAS, which is really not very promising, but we often target downstream. So because we know that once RAS is activated, it's going to activate its downstream pathway, which includes like MEK, ERK, and other things like PI3K. So what we can do is we can target those things. And I need to tell you that those things are kinases, right? So then we can synthesize kinase inhibitors to target those downstream. But now, and this is work from actually Kevin Shoka, so one of our collaborators, and he actually and others, they've actually started to synthesize specific KRAS inhibitors. In the past, people used to think that KRAS can be targeted, right? So but now they've actually come up with a very novel chemistry idea and powerful thinking to come up with inhibitors that can directly target KRAS to reduce its oncogenic activity.
Okay, thank you so much for that explanation. I hope it's actually answered the questions that we have. The next question said, for all these genes and pathways involved in cancer, it's becoming obvious that we have a long way to go to end cancer. What's your take, sir?
Yeah, this is a very, very good question, and I'm pretty sure there are better people that can, that can answer this more than myself. But what I'm thinking, based off of my, little understanding, is that so cancer is a complex disease, and requires multiple pathway. So rather than targeting just one pathway, so we can't just target KRAS pathway, right? So we need to find a way to target multiple pathways. So, and that is where, for example, model system will come in, that they can allow us to target different types of things, and then we can come up with compounds that will target different arms of the disease. So I think the future is that we need to always think of multiple pathways rather than single. In addition to that, we need to think of compounds that will prevent toxicity. So when you go into the history of cancer therapeutics, in the 1940s, people started using surgery and radiation, moved to chemotherapy. From the 2000s, people started using targeted therapy. And now we've got different types of compound, different types of really drug family that people are using now, including gene therapy, protax, and immunotherapy, right? So, but a lot of these type of compounds, they're still focusing on one target. Oh, my immunotherapy will focus on EGFR inhibitor. Oh, I'm going to focus on this type of inhibitor. So, but we need to start thinking of multiple things, right? So rather than just one, I think that would be the future. And I'll say, now I think the reason why we are struggling in the past is because we don't have the right computational tool to do this. But now I think with the likes of artificial intelligence, improve in science and improve in research, hopefully more money, we can begin to think of ways to actually tackle these multiple networks in cancer, and hopefully come up with better drugs and better treatment for patients.
Thank you, doctor. And for the future we're talking about, can we say that Drosophila is enough? Can it be used for all? Because when they talk about replicating it, using it as a model because it has over like 75% similarities, can we say that 15% that's not there is not enough to destroy the good work that is happening? Is it enough? Can it be used for other um, more kind of severe cancer symptoms coming up now?
No, this is a very, very good question, and this is a very good question. Thank you so much for that. So yeah, so I would say, like my boss used to say, so Ross Kagan is like, because something quack like a duck doesn't make it a duck. That's his word for word. So yeah, so because something can represent a patient doesn't make it a patient. So far I can tell you for a fact that there is no good model. We've only got useful models, right? So there is no model that will be able to answer all the questions. So the only person that can answer my own question is me. The only person that can be used to understand my disease is me. Is that okay? So there is no other person, there is no other model that will be able to do that. But we cannot use me for research. So, and that is where our models comes in. So to me, Drosophila will continue to be useful for this type of research, including cancer, including other types of biological process. Drosophila will continue to be useful, but we need to be cautious and we need to be conscious of how we are interpreting our data. And also, your goal as a scientist is to, you need to make sure that you've got multiple things that you can test your let's say your drug of interest on, and also collaborate. So, I mean, there's an example of our collaboration with Amos, right? So you guys, you can also have collaborators in, let's say, outside your environment, or people that have got things that you don't have. Is that okay? So then you can build on that. So you can test your compounds on flies and be like, okay, these people have got zebrafish model. Can we test on it? Or you've tested your extracts on flies? Those I've got zebrafish model or mouse model. Can we test on? So these are things that that you can also use to help your own research. But in summary, Drosophila will continue to be useful, but we need to always try and check what we're finding in other models as well.
Okay, so then what you're saying is that there is room for using combined and use other models alongside the Drosophila. Thanks very much. Our time well fast spent. So with that, we take one last question. And it says sequencing is very expensive in African setting. Do we have a means of subsidizing the cost to enable researchers in this part of the world carry out elaborate research like this one?
Yeah, this is a very good question, and I would reiterate that research equals money. So you need money to do proper research, honestly. So the beauty of the future, and even right now, not as even from now, is that things are becoming really cheap. And also with what you guys are doing, with what others are doing, researchers from from, let's say, from the western part of the world, they're beginning to open up to collaborating with, for example, people in like West Africa, or like in Africa as a whole. There's opening for that, right? So even if you don't have that cost, by building an important collaboration with someone that knows that you're serious with what you're doing, you have to lead with that seriousness. Then the cost might not matter, because they're going to be happy to sequence things for you, and then you can use that data to up your own research, because they are also going to benefit from that data, right? So but to me, that would be one solution, the first solution. The second solution to that is that, you know what, sequencing is going to continue to be cheap. So in the past, sequencing a sample can be 10K, but now is about £300. So, it is really, there was a cut down in cost, and because people have made their money already on them, and now they're just subsidizing things, and they want more people to use it, and then I think things will become really cheap. But having collaborators outside your own environment will be very, very important. And it can be difficult to find these collaborators, because I'll tell you that it can be difficult, but honestly, I'd advise that you start. Is that okay? And also talk to your policy people as well, because I mean, in Africa, I think, to me, there was a lot of money there. People are spending on things that to me might not be useful, but talk to them as well. Convince them that these are very important. Convince them that we can improve that part of the world. And then I think, from there, things that we thought are really expensive becomes really cheap.
Thank you to heard about collaboration, and thank you for addressing the part of very difficult to find collaborations, because you might be having one part of your research and the other person has the second part. Coming together will actually help the work to be very. So, in your closing remarks, can you just an advice on limitations that researchers should be looking forward to, and to overcome these limitations, these barriers that are in the setting, the African setting as a whole?
Yeah, so I mean, I've got really good relationship with with people in Nigeria, and especially in the research part of things. So to me, I think comparing, because I mean, I was trained as a researcher in this part of the world. So I'd say, based on my training and based off of what I'm seeing as training over there, is people are really confident over there, not unlike in the past. I think things are changing, people are really confident. So, but I'm worried about the influx of AI, that I think what I've learned is, people are not using this tool as they should use them. So to me, that's one limitation that I think people should work on. So, and also I think there is really limited understanding about what people are doing as well. So I think I'm worried that people are not reading as much. So I think reading as much as possible to understand the concept of what you're doing, and actually finding proper evidence. Because you can prove me wrong, that's fine. I'm not I'm not the island of the knowledge. So that, to me, that's really what we need to start bringing into our early career research. And now that that as a professor, you don't have the final say about this topic. Your students should be able to be like, actually, we think that is not correct. And not necessarily in a rude way. You have to be smart about the way you talk to people, but be able to communicate that actually, I can be wrong, you can be right. And then I think that would be one way for us to move things forward in that part of the world. In addition to that, is we need to be selfless as well, and we need to be understanding of each other. That okay, this person have got their eyes that today, tomorrow they can have their lows based on personal reason. So we need to understand that people differences as well. So, and in terms of I mean, that is dealing with people really and as individual. So to me, in terms of the grants and the finances that will come with that limitation, is honestly, I think we need to start talking to our policymaker in like properly, and we need to then be a good representation of research. Because, for example, people are happy to spend money on, let's say, music, for example, because they can see that, oh, people love music, right? So I think as scientists, we need to be a good advocate of what we do, and talk to people about this. Talk to your family member about this. Let them understand what you're doing. Let them understand the impact of what you're doing. Show them evidence of the impact that have been, that people have shown, for example, in other countries. Okay. So, and then I think, Africans are quite competitive. So if this is happening in one part of Africa, I'm pretty sure that other part of Africa will want to emulate it. So we just need to be good examples for for one another, so that we can improve. We can improve our science. We can improve our standard of living. We can improve our humanity. We can improve our mental health, and we can improve our overall life.
Thank you so much, Dr. Badmos. Thank you so much, everyone, for your active participation, for the questions that you brought this way. We want to appreciate the presence of the Director of Research and Innovation of the African Center for Disease Control and Prevention, CDC, Dr. Musoka Papafala. Thank you so much for joining us today. We appreciate you. So today's session has been very interesting. It has showed us the fundamental research going on using Drosophila and the contributions it has had to cancer research. He has also talked about the limitations, and as we all go back to our various settings, Nigeria, Rwanda, Africa, different countries that we are, we look out for collaboration. That is the key word, because we have several limitations, we have several barriers to our research work. Collaboration will help us grow, open us to new idea, open us to opportunities, especially funding and grants that can help push research forward. Thank you so much, Install Africa, for giving us this platform to learn more. To the students, to the professionals, to the researchers, to the administrative staffs, we say thank you. So this is FIRAT. We hold events every month. We have the mental health platform, maternal health, dental oral. We have the clinical platform. Every platform you want, you can go on YouTube, follow us on YouTube on FIRAT, and you can go back to our recordings to remind yourself of so many things that have been there, or you want to learn more, you can watch it. If you haven't registered, you can register with us, and if you have, you'll be receiving more information in your mail. So thank you so much, everyone, for today. Have a wonderful day, and we hope to see you next month in our next webinar session. Thank you.
All right. Thank you, everyone, and have a good day.
Same to you.
All right.
Bye.
All right. Thank you. Bye. Mhm.